Okay, so my name is Sarah, and I will be presenting the last session of this webinar, and I will be discussing colony collapse, pesticides, and honeybees. So this is a brief outline of my talk. I will go over colony collapse disorder, as well as pesticide exposure of honeybees, current pesticide risk assessment procedures, and potential future pesticide risk assessment with a focus on neonicotinoids and honeybees. So, colony collapse disorder. This is a phenomenon which appeared in the winter of 2006 to 2007 in the United States, where commercial beekeepers were reporting large-scale losses of honeybee colonies. And this colony collapse was characterized by sudden disappearance of worker honeybees from the colony, with no apparent dead worker bees around these affected colonies. The bees appeared to abandon their brood and food stores, and the disappearance was quite mysterious. So occurrence of these colony losses caused beekeepers to look back in history to see if this was the first time that colony collapse had occurred. And in fact, colony collapse disorder is not a new phenomenon. Since 1868, beekeepers have been reporting large-scale colony losses episodically, both in the United States and elsewhere in the world. And alarmingly, colony losses have continued at an unsustainable level. So since 2006, 2007, the United States has kept records of colony loss, and it has reached as high as 43.7% last year were the reported annual colony losses, which is the second highest on record. On average, annual colony losses since 2010 have been approximately 39% in the United States. And so for a commercial beekeeper, losing 40% of his or her colonies could represent a significant economic burden. Similarly, in Canada, we have also observed increased colony losses, and in particular, we monitor overwinter colony loss. And in general, winter colony loss of 15% is considered long-term sustainable for a beekeeper. And since 2007, Canadian beekeepers have been experiencing colony losses in excess of this 15% threshold, with a 30% loss since in the winter of 2020. In spite of these losses, we have seen that the number of colonies in Canada has gradually increased. So the gradual increase in colonies despite colony loss speaks to the resilience of beekeepers and their ability to split their surviving stock in order to multiply their colonies. So what is the cause of these colony losses? Well, in general, there has been no single stressor that has been identified. And the general consensus is that it is interacting stressors which are precipitating colony loss, and that the cause is multifactorial. So many times during the seminar, you have heard the presenters speak about the host, pathogen, and environmental determinants of disease. And so it is no different in this case when we speak about colony collapse, in that there are various host, pathogen, and environmental stressors which are interacting to result in colony loss. And we can think of the host and pathogen sort of on a teeter-totter, with the environment bringing the host and pathogen in contact with each other and potentially tipping the balance in favor of one or the other. So For example, if we have a positive environment where colonies are provided with clean water and food, diverse forage, and the colonies are well insulated and well ventilated, then this kind of environment may favor the host and decrease the host's susceptibility to disease and improve the host's immunity. In contrast, if we have negative environmental stressors, such as pesticides, poor nutrition, or transportation associated with migratory beekeeping, these stressors can tip the balance in the favor of the pathogen, resulting in decreased immunity of the host and increased susceptibility to disease and potential colony collapse. So for the remainder of this talk, I'm going to focus on pesticides as a potential environmental stressor and how pesticides could be contributing to colony collapse. So what is the exposure of honeybees to pesticides? Well, in 2010, Mullen et al. Set out to survey the prevalence of pesticide residues within honeybee colonies in North America. And what these researchers found is that pesticide detection was extremely common and varied. Approximately 121 different pesticides were detected within North American honeybee colonies. And greater than 90% of the bee, pollen, and wax samples that they collected had more than 2 pesticides per sample, with an average of 6 pesticides detected per sample of bees, pollen, or wax. So what are these pesticides that honeybees are being exposed to in the colony? Well, the top 5 are listed here. And interestingly, 3 of the top 5 are miticides that beekeepers are applying themselves. So common pesticides that honeybees are exposed to are in fact pesticides that beekeepers are using. However, there is also a variety of agrochemicals which were detected. So chlorpyrifos and chlorothalonil, which are listed here, are an insecticide and a fungicide, respectively, which are used in commercial agriculture and are common contaminants of honeybee colonies in North America. So how are honeybees being exposed to agrochemicals? Well, there are a number of routes. There can be direct exposure through foliar spray of pesticides directly on foraging honeybees. However, this has been minimized in recent years due to good communication between beekeepers and farmers and application of foliar sprays during the early morning or late evening when honeybees are unlikely to be out foraging. Honeybees can also be exposed to the dust generated during planting of seeds treated with agrochemicals. However, once again, farmers have modified their planting procedures to minimize dust and exposure of honeybees. Honeybees can also be indirectly exposed to agrochemicals through routes such as contaminated soil or water. But probably one of the most concerning avenues for exposure is exposure to pesticide residues during foraging on pesticide-treated crops. And in particular, the systemic pesticides have been implicated as a source of residues for honeybees who are foraging on these crops. And neonicotinoids are probably one of the most famous and controversial examples of a systemic pesticide that is widely used today. So these insecticides were introduced approximately 30 years ago, and they have rapidly grown to be one of the most popular classes of neuroactive insecticides used worldwide. One of the reasons for their popularity is their high safety, and these insecticides are highly selective for the nicotinic acetylcholine receptors of insects. and are not extremely toxic to mammals or vertebrates, giving them a high safety for humans. The three most commonly used neonicotinoid insecticides for outdoor agricultural uses are clothianidin, thimethoxam, and imidacloprid. And here on the Canadian prairies, greater than 95% of our canola is grown from seed that is treated with a neonicotinoid. So as I mentioned, neonicotinoids are applied as a seed treatment. So when these treated seeds are planted in the soil, the insecticide is water soluble and it is taken up into the growing tissues of the plant, where it will then be present in low concentrations within the pollen and nectar of that plant, presenting a route of exposure for a honeybee foraging on that crop. Neonicotinoids are neuroactive insecticides, so they act on the central nervous system of exposed insects. And so this diagram here represents a synapse within the central nervous system of an insect. And so the neonicotinoid will bind irreversibly to the postsynaptic nicotinic acetylcholine receptors, resulting in sustained stimulation of that postsynaptic neuron. And this can result in neurologic clinical signs and at high enough concentrations, death of the insect. But in most cases, we know that neonicotinoid exposure in the environment is sublethal for honeybees. So individual honeybees foraging for a several kilometer radius around their colony may bring back these contaminated food stores, nectar and pollen, to their colony. But the interesting thing about honeybees is that individual exposure to pesticides also results in colony level exposure. And so there is potential for pesticide bioaccumulation within a colony. As I mentioned, these foraging honeybees are foraging for a several kilometer radius, so they can pick up residues from a wide area, and then these pesticide residues are then concentrated within the hive into honey and bee bread. Furthermore, this honey and bee bread is subsequently fed to the colony's brood and bees. And so through this process, we can have accumulation of the residues. Furthermore, we know that pesticide residues can last for up to years within the wax of comb within the hive, presenting a continual source of pesticide exposure for the colony. So we know that honeybees are being exposed to pesticides. How do they process and how do they cope with these pesticide residues? Well, typically they're going to use a variety of metabolic enzymes to process and detoxify these pesticide residues. However, unfortunately, it appears that honeybees are disadvantaged when it comes to metabolic detoxification of pesticide residues. So three of the biggest classes of detoxification enzymes in insects are listed here in this table. And when we look at the honeybee genome and compare the genome to other insects such as fruit flies or mosquitoes, we notice that honeybees have far fewer genes for these detoxification enzymes compared to other insects. So honeybees have half as many genes for cytochrome P450 monooxygenase enzymes compared to fruit flies or mosquitoes. And thus, we're concerned about whether or not honeybees will be able to detoxify pesticides as effectively as fruit flies or mosquitoes. Similarly, for other detoxification enzymes such as glutathione transferase or cholinesterase, honeybees have far fewer genes compared to fruit flies and mosquitoes. So we wonder whether or not honeybees can cope with this long high levels of pesticide exposure in the hive and whether our continued use of agrochemicals will be sustainable. So ultimately, what we need to determine is the safe dose range for pesticides and honeybees, because we know that we need to use agrochemicals to keep our crops healthy, but at the same time, we need healthy honeybees with which to pollinate these crops. So oftentimes in pesticide toxicology, we talk about the dose makes the poison. And so here in this graph, we have effect on the y-axis, and we have dose of the pesticide on the x-axis. And as the dose increases, the severity of the effect will increase. So at very high doses of a pesticide, we might expect severe effects on a honeybee colony. So for example, acute death of all of the bees in a colony. However, at more mid-range doses of that pesticide, we might expect more sublethal effects. So these effects will not be as obvious and could include things like abnormal behavior or increased disease within a colony. But ideally, we would like to keep the dose of pesticides within the safe dose range. So low doses which have no observable effect on exposed bees. So how do we identify what the safe dose range is? Well, in most cases, we conduct a pesticide risk assessment. And so these risk assessments are usually conducted by regulatory agencies, such as the United States Environmental Protection Agency, or here in Canada, the Pest Management Regulatory Agency. And so the first phase of a risk assessment is to establish whether honeybees are being exposed to the pesticide in the environment. And if the answer is yes, then the regulator will conduct a tiered risk assessment. So the first tier of pesticide risk assessment involves laboratory experiments where adult worker honeybees or brood are acutely or chronically exposed to pesticides to determine the toxic effects. And in determining the median lethal dose, or LD50, the dose which kills 50% of the exposed bees, we can then evaluate the risk to those individual bees or brood. And based on the assessment of risk, the regulator can then decide whether or not the risk is acceptable. And if the risk is deemed to be unacceptable based on these laboratory experiments, then the risk assessment will proceed to the second tier. And these tier 2 semi-field experiments involve exposure of adult bees to a treated crop in a mesh tunnel or cage. And so in these tier 2 experiments, the regulator is better able to assess colony level effects of the pesticide on mortality of the bees. However, it's still not a truly realistic situation. So if based on tier 2 trials, again, if the regulator deems that risk still remains high, then the assessment will proceed to tier 3 trials. And so these are full field experiments using free-flying honeybee colonies, which are allowed to forage on treated crops. And so tier 3 trials allow for the most realistic assessment of colony level effects, including observation of foraging behavior and production of brood, as well as pesticide residues in the hive. As you can imagine, as we move from tier 1 through to tier 3, the realistic nature of the assessment is enhanced. However, the expense and complexity of the risk assessment is also enhanced as we move from tier 1 to tier 3. And not all pesticides will go through all 3 tiers of risk assessment if the tier 1 risk assessment deems the risk to be manageable. So no risk assessment is perfect, and there's always room for improvement. And so one of the focuses of our lab's research is to develop new tools for pesticide risk assessment, which can perhaps enhance the regulatory decisions regarding pesticides. And so one of the important things to remember when thinking about risk assessment is that honeybees are a eusocial species. And so just to recall what a eusocial species is, so this means that this species has division of labor, has division of individuals into reproductive and non-reproductive individuals, and that these individuals cooperate in order to care for the colony's brood. And within that colony, there will be overlapping generations of parents and offspring. And so this is a very specialized organization, and lends itself to a superorganism where the colony is perhaps the true individual, and that the bees within the colony are just part of a greater whole. And so if we think about honeybees as a superorganism, and then we can imagine that pesticide risk assessment may be need to be tailored to account for the various eusocial biology of honeybees. So in particular, in our lab, we are starting to do specialized pesticide risk assessment for particular castes within the colony. And so Dr. Ivana Cozy in our lab has led the development of histopathology of the queen reproductive tract as a method for assessing pesticide exposure in honeybee queens. As well, Dr. Colby Klein in our lab is looking at honeybee drone reproductive toxicology with regard to neonicotinoid insecticides. And again, thinking about the specialized role that drones play within that superorganism and how pesticides may differentially affect this caste of bees. And so we believe that these new avenues for pesticide risk assessment are incredibly important because pesticides are so controversial in society and the media today. And so this controversy has lent itself to various and often conflicting regulatory decisions regarding the use of pesticides. So for neonicotinoids, currently these pesticides are banned in the European Union and have been so since 2013. While in contrast, here in Canada and the United States, neonicotinoids continue to be used by farmers. Although the use of neonicotinoids is currently under review by Health Canada. And so considering the often conflicting decisions made regarding pesticide regulation, there is need for increased scientific evidence to support these regulatory decisions. And so here in Canada, we wanted to ask the question, what is the safe dose range for neonicotinoids and honeybees in our local area of Saskatchewan, Canada? And could we design a new pesticide risk assessment which might inform some regulatory decisions surrounding neonicotinoids? So as I mentioned, neonicotinoids are used widely in our area of Canada, and it's not surprising that they are commonly detected in honey from our area. So this graph here shows the concentration of neonicotinoids in nanograms per gram on the y-axis in 30 different samples of honey on the x-axis. And the bars show the concentration of various neonicotinoids. And so we can see from this graph that the sort of upper limit of high environmental concentrations of neonicotinoids in Saskatchewan honey is approximately 20 nanograms per gram, although there are some detections in excess of this threshold. But the more mid-range concentration of neonicotinoids in Saskatchewan honey is around 5 nanograms per gram. So we were wondering, how do residues in Saskatchewan honey compare to neonicotinoid residues worldwide? And so for the neonicotinoid thiamethoxam, the average concentration in honey worldwide is approximately 0.3 nanograms per gram. In contrast, in Saskatchewan honey, the average concentration is 17 nanograms per gram. So this is 57 times greater the global average. And so our lab wanted to investigate what could be the effects of chronic exposure to these high concentrations of neonicotinoids in honeybees, and what could be the consequences for our honeybee colonies. In particular, we wanted to look at the effects on colonies which might be more vulnerable. So nucleus colonies or small colonies were one subset of colonies that we wanted to assess risk for. As well, we wanted to look at the effects of chronic neonicotinoid exposure on overwintering colonies, which also may be vulnerable to pesticide stress. And thirdly, we wanted to look at the effects of neonicotinoids on worker brood, which again may be vulnerable due to their young age. So to begin, we'll discuss the effects on nucleus or small colonies. So our hypothesis was that chronic exposure of small nucleus honeybee colonies to neonicotinoids would decrease their colony performance. And we wanted to test this hypothesis using mid-range environmental concentration in Saskatchewan of 5 nanograms per gram and a high environmental concentration of 20 nanograms per gram. And so what we found is that small colonies that are chronically exposed to high concentrations of neonicotinoids have a decrease in honey production. So this graph here shows colony weight gain on the y-axis and time on the x-axis. And the bars represent different concentrations of neonicotinoids to which these colonies were exposed. So the white bars are the control, and the gray and blue bars represent colonies which were chronically exposed to neonicotinoids. And what we can see is that after 9 and 12 weeks of chronic exposure to neonicotinoids, that these small colonies gained on average 30% less weight relative to controls. And so weight gain is primarily influenced by honey production of the colony. So on average, colonies exposed to 20 nanograms per gram of neonicotinoids produced 30% less honey relative to controls. However, interestingly, the colonies exposed to 5 nanograms per gram neonicotinoids did not have significantly different honey production relative to controls. And so what this tells us is that chronic high concentrations of neonicotinoids can have negative impacts on honey production of colonies, and that honey production may be a useful outcome measure for pesticide risk assessment. Furthermore, we wanted to investigate the effects of chronic neonicotinoid exposure on overwintering colonies. So we know that here in Canada, winter is long, and that there is potential for chronic exposure if these colonies are relying on honey and bee bread, which is contaminated with neonicotinoids. And our hypothesis was that chronic overwinter exposure to neonicotinoids will decrease the survival of colonies. And specifically, we tested this hypothesis using two concentrations. So 20 nanograms per gram, which is a high environmental concentration of neonicotinoids, and 100 nanograms per gram, which is an unrealistic concentration for the environment, but was used as a positive control. And so what we found is that only at unrealistic concentrations does chronic neonicotinoid exposure decrease overwinter survival. So this graph here shows percent winter mortality of colonies on the y-axis, and the x-axis shows treatment group. So at high environmentally unrealistic doses of neonicotinoids, colonies experienced a significant 55% increase in overwinter mortality relative to the control. But at environmentally realistic concentrations of 20 nanograms per gram, there was only a 15% increase in overwinter mortality relative to control. And so this was not significantly different from the control. So what this tells us is that chronic overwinter exposure of honeybee colonies to neonicotinoids would not significantly impact overwinter survival at environmental concentrations. And thus, the risk to overwintering colonies from neonicotinoids is likely acceptable. Our last objective was to investigate the effects of chronic neonicotinoid exposure on worker brood. And so, as we've mentioned during this seminar, it is possible to raise honeybee larvae to a pupa and then an adult within an incubator in the lab. And so we exposed these larvae to neonicotinoids and fungicides chronically during development and looked at their effects. So our hypothesis was that honeybee larvae will have increased mortality and increased susceptibility to disease when they are chronically exposed to neonicotinoids, fungicides, or the combination of neonicotinoids and fungicides. And so this graph here is showing brood survival on the y-axis and then treatment group on the x-axis. So the red bar here shows the survival of our control larva from larva into adult. And the other bars represent pesticide-exposed larvae. And so we can see that at high concentrations of neonicotinoids in combination with a fungicide, so 500 to 250 times environmental concentrations of neonicotinoids, we did have a significant decrease in survival of honeybee brood. However, for larvae that were exposed to lower concentrations of neonicotinoids with a fungicide, so from 100 to 5 times environmental concentrations, we did not see a significant decrease in brood survival to adulthood. And so what this tells us is that at environmental concentrations of neonicotinoids, in combination with a fungicide, we would not expect a negative effect on brood survival. However, just because there's not a direct effect of pesticide exposure on survival of brood does not mean that there could potentially be sublethal effects of this chronic pesticide exposure. So as we've mentioned before, in our lab, we are able to infect honeybee larvae with a bacteria called Melissococcus plutonius, resulting in replication of the brood disease European foul brood. So we can use this model of disease to investigate whether pesticide exposure may enhance susceptibility to European foul brood. And so this graph here is showing larval survival on the y-axis and infectious dose of bacteria on the x-axis. The colored bars here are representing treatment group. So The dotted bars are the control group. The red bars are the fungicide exposed group. And the blue bars are the group exposed to a fungicide and a neonicotinoid. And so I'll draw your attention here to the group that were infected with 50 bacteria. And what we see is that the larvae which were exposed to a neonicotinoid in combination with a fungicide had a significant 25% decrease in survival relative to infected controls. So this data might suggest that those pesticide-exposed larvae were more susceptible to bacterial infection and disease compared to controls. However, the important thing to mention here is that the concentrations of neonicotinoids we used were 84 times those which would be considered environmentally realistic. So if we really want to understand the effects of pesticides on susceptibility to disease, we would need to repeat this with more realistic concentrations of neonicotinoid. So in conclusion, based on this research, what can we say about the safe dose range for neonicotinoids and honeybees in Saskatchewan, Canada? Well, based on our work, we would suggest that the threshold for harmful effects on honeybees lies around 20 nanograms per gram. And that in order to maintain the health of our colonies and maintain their honey production, we need to keep residue levels below 20 nanograms per gram in order for our crops and our honeybees to thrive. And so I hope by discussing some of this research, I can give you an example of how pesticide risk assessment can be modified and tailored to particular regions in order to assess risk to pollinators. And so just to wrap up this talk, we discussed colony collapse and how the etiology of colony collapse is multifactorial, that there are many interacting stressors which are responsible for increased colony losses. And pesticides may be one of these stressors because we know that pollinators are being chronically exposed to combinations of pesticides, both in the environment and within the hive. And in order to assess risk of these pesticides to honeybees, we need to consider the honeybee as a superorganism and tailor our pesticide risk assessment accordingly. So with that, I will thank our funding and my colleagues in the honeybee lab, and I would be happy to take any questions. Okay, so there's a good question here from Lloyd, who wonders if LD50 testing of pesticides is done on adult bees and no testing of effects on brood or other casts, what can be done to resolve these issues? So basically, Lloyd is getting at the issue that oftentimes doing pesticide risk assessment on adult workers is not adequately reflective of the different ages and different castes within a colony. And so you're completely right that we need to take a broader view of pesticide risk assessment. And one of the avenues to improving risk assessment is to conduct research, because regulators do look at the literature to inform their risk assessment. And if, for example, we're consistently showing negative effects of pesticides on honeybee queens, then regulators will potentially take that into account and may modify their risk assessment in the future. All right, so we have another question here from an anonymous attendee asking if honeybee colonies are on the rebound, are they using the same pesticides or different pesticides than the United States? And so I would say that honeybee colonies are not necessarily on the rebound, even though there's a gradual trend toward increasing numbers of colonies. We're still experiencing severe overwinter losses, which are not sustainable for beekeepers in Canada. And so I don't know that we're much better off here in Canada, although we may have fewer migratory beekeepers here in Canada, which might buffer some of our colony losses. So I don't know that we could say that pesticide use is that different between the United States and Canada. Neonicotinoids are widely used in both countries. And so I'm not sure we could explain differences in colony losses based on pesticide use in the two countries. Okay, and then we have a question from Alex asking, what are some of the overlooked environmental factors that can be harmful to a hive when determining the location of a hive? Is proximity to a power plant, wastewater, or factory dangerous? So certainly there's other environmental stressors, there's heavy metals in the environments, there's all kinds of contaminants which honeybees can potentially encounter when foraging. And so it's really hard to necessarily know what is the best location. But certainly I think you'd want to take those things into consideration. If there is a source of contaminants close to within, a 5-kilometer radius of your hive, then there's a good chance that some of those contaminants may be coming back to the honey and pollen that is present within your colony. Okay, and so Janice asks, does Saskatchewan have a maximum rate for nanograms per gram used? So to my knowledge, at this time, we do not have necessarily exact number for pesticide concentration, but neonicotinoid use is currently under review by the Pest Management Regulatory Agency, and so I think some of these threshold levels are being established. And certainly they're looking at the levels of neonicotinoid contamination in fresh water and establishing, a safe dose range for not only honeybees, but aquatic invertebrates as well. And certainly, pesticide use is going to vary depending on the area of the province. So we have some more agriculturally intense areas of Saskatchewan and some more wilderness areas. So accordingly, pesticide residues are going to vary depending on the level of agriculture. And then we had another question from David asking, he is curious about the conclusion between 5 and 20 nanograms per gram as not significant. It appeared that many losses were double the 5 nanogram. Can you comment? Well, I mean, sometimes there can be, for example, a negative effect of pesticide exposure, but it may not necessarily be statistically significant. And so I think there was another question as well relating to statistical power. So sometimes when we do these studies, we may, for example, see a decrease in overwinter survival, but it's not significantly different from control. And this could be because there is no difference from control, or it could be because we did not have enough statistical power to detect an effect. And so sometimes in these field studies, you need a large number of colonies in order to detect a significant effect of the applied pesticide. And so because of the labor involved in managing many colonies within a toxicology study, sometimes it's just not possible to have adequate numbers in order to have statistically significant effects. So we do the best that we can as researchers, but I guess, as a member of the public or a beekeeper, you do need to take all studies with a grain of salt. And remember that these studies were conducted on a small population under controlled circumstances, and it's not always reflective of the real world scenario. Okay, so I think I will stop it there, and perhaps I'll turn it over to Elamir if he has any concluding comments.
No, I don't have any concluding comments. I thank you again to Davis-Thompson Foundation for organizing these. I thank you to presenters for this marathon. A great job. I see that I should have assigned my presentations to them, but I had to have mercy, for example, on Ivana. She's preparing for her comprehensive exam. She'll have it in a couple of weeks, and she still took at least part in it. So anyway, thank you to everybody. Thank you to your interest. And yes, Sarah summarized very well, specifically these pesticides. It is, yeah, is it significant difference between control that is 10% mortality and 5 nanograms, 25% mortality? Well, obviously, it is a big deal. If you have 100 hives, whether you have 15 hives more dead. Yes, it is. But when you look at the scientific statistical analysis, we have to work on that. And in order to confirm change, we have to be within statistical significance. And so if we have statistical significance, we say that, yes, there is difference. If we don't, it doesn't mean that there is no difference. It just means that we did not detect with the experimental design that we used. Anyway, thank you. And does moderator want to do any concluding remarks?
Yes, if no one minds. Paco, do you want to say a few words before I do my final announcements, or?
Okay. Yes. Thank you, Eileen. Thank you for moderating too. Hey, Elamir and everyone. Thank you very much. This was a fantastic seminar. At some point, you had close to 1,000 attendees. It's a completely new topic for the foundation, clearly of great interest to everyone. And more importantly, I like what you said at the beginning, Elamir. You started from the very, very basics that maybe not for other people, but for me, it was great. I really thank you for that. And let's hope this is not the last one. You guys have opened the door to what I think will be more activities of the foundation on this topic. So thank you again and congratulations, everyone.
Thank you. Thank you in the name of entire lab.
Thank you. Over to you, Eileen.
All right. Well, I just want to reiterate again, thank you so much to our many presenters today, Dr. Simko, Zabrodsky, Cozy, Wood, Medici, Klein, and Thebu, for this absolutely amazing full-day seminar on honeybee diseases. I hope you take a moment to look at the chat comments because people have been raving about the talks all day long. This was absolutely incredible, and we hope that all of our participants enjoyed today's seminar. Today's lectures will be available for a short period of time on the Foundation's YouTube channel, either later today or within the next couple days. I know several people had questions regarding this over the course of the day.